Hello, my name's Annie. My research today is about the phylogenetic relationships of arenaviruses in Neotomine rodents, a test of host specificity. The arenavirus is a rodent-borne virus in the family Arenaviridae. It's a spherical, enveloped, ambiscent, single-stranded RNA virus with two segments, the L and the S segment. The S segment is composed of the glycoprotein and the nucleocapsid gene. The L segment is composed of the RNA-dependent RNA polymerase and the ring domain. In our research today, we are focusing on the N gene. The natural hosts of the arenaviruses are species of rodents, specifically neotomine rodents. It's important to note that not all species of Neotoma have been documented to be hosts of arenaviruses. There are currently 16 different strains of arenaviruses that exist in North America, 11 of which were examined in this study using the host Neotoma species, are in bold on my poster. North American arenaviruses are generally considered non-pathogenic, but some of these viruses have been implicated in human fatalities. There are 7 arenaviruses in South America and 3 arenaviruses in Africa that cause disease. In our picture here, here's a picture of a Sigmodon hispidus and 3 other Neotoma species. Our objective today is to identify the phylogenetic relationship between North American arenaviruses strains that occur in Neotoma and Sigmodon, to characterize the genetic divergence among and within North American arenavirus strains. Again, we took a region of the S segment, specifically the N gene. And our last objective is to examine host specificity between the phylogeny of the host Neotomine rodents and the phylogeny of the zoonotic vector arenaviruses by comparing their phylogeny. Materials and methods. Obtain all S segments and N gene sequences associated with arenaviruses on GenBank. To visualize and trim the sequence data to only include the N gene. We also align the sequence to be in the correct reading frame. We filtered for arenaviruses strains that had Neotoma as a natural host. An exception are the Tamiami virus, in which their host is the Sigmodon. We then used RAxML to identify and remove identical sequences that were 100% similar. We then identify the model of nucleotide substitution. Then we generated a Bayesian phylogenetic analysis using Mr. Bayes, generated genetic distance between and within 10 genera of arenaviruses. And lastly, we compared the 2 trees between hosts and arenaviruses, rejecting the host specificity. Here are our results. In Figure 1 demonstrates the phylogenetic relationships of all documented arenaviruses and wild-caught neotomine rodents. 16 of the 20 nodes exhibit posterior probability values above 0.95, meaning that it's Bayesian statistically supported. Figure 2 shows the phylogenetic relationship of neotomine rodents using a combination of 9 molecular markers, 2 nuclear and 2 mitochondrial, with various Bayesian and maximum likelihood supported throughout the tree. We were able to show that the majority of arenaviruses relationship is supported because viruses evolve so quickly. This is a huge accomplishment. Table 1 displays the genetic divergence within and among different strains of arenaviruses. Within the strains, the lowest is 0.14% in the Palo Verde virus, the highest is 15.04% in the Tamiami virus. Among strains, genetic divergence range from 18 to 31%. The genetic distance rate is so high because they evolve so rapidly and have a faster generation time, and they're in a constant arms race with host neotomas receptors. Table 2 demonstrates the species, what virus has been detected, and their geographic distribution. Neotoma albicula is the host to 4 arenaviruses, Mexicana and Micropus to 3, and Leucodon to 2. This relationship is shown on a map in Figure 3. For our discussion, a comprehensive phylogeny of North American Arenaviruses is presented for the first time. Comparison of the virus phylogeny to the host phylogeny provides evidence that can be used to reject host specificity. For example, 3 woodrat species are host to multiple Arenaviruses species. One Arenavirus occurs in 3 different woodrat species, and a second Arenavirus occurs in 2 woodrat species. These data indicate that Arenaviruses may occupy a distinct geographical area and infect one or more woodrat hosts that occupy that region. For our future direction, it's to construct tangograms to further examine in detail the presence or absence of host specificity between Neotomine rodent hosts and Arenaviruses using the program tree map. Potentially increase the sample size for several Arenaviruses, and determine additional host-virus interactions in associated ecosystems like ecological impact and implications for climate change. And lastly, a thank you for everyone that's provided support and edits to this poster.